All right. Okay, so we're gonna start our session about different topics in biology. And our first presenter is gonna be Emily Rodriguez Castello. She's joining us from Wayne Community College. She's an honors student there and a dual enrolled student, and she's pursuing an associate in science degree. Her paper is concerned with the growth and loss trends of the Anaxyrus californicus. And I look very much forward to your talk.
Thank you. So as said, my paper is on the growth and loss trends of the Anaxyrus californicus, specifically using iNaturalist as a research tool. Okay, so before I wanted to actually go into my hypothesis and my question, I wanted to talk about what citizen science is and what iNaturalist is, because going into this research, I didn't know what either of those things were. And I feel that we should, I guess, increase awareness of citizen science and sites like these to expand our knowledge and involve the community in science and science projects. So citizen science is the practice of public participation in scientific knowledge to increase scientific knowledge. And through citizen science, people share, contribute data monitoring and collection programs. And this is what iNaturalist is centered about. It's a citizen science initiative. It's a free platform. And you record observations of plants and animals in nature. You can submit pictures, when and where you found this organism. And tools with iNaturalist can help you identify what this organism is, or the community can actually find out what this is. And this all kind of is like a database, and you can use information found by other people to research organisms like I did. Yeah, it's a data portal. So my paper centered around the Anaxyrus californicus. This is the commonly known as the Arroyo Toad, and it's a species of toads found in California and in places in Mexico. And I chose this organism because it's currently classified as endangered. And I thought it would be interesting to see how species numbers would be reflected in iNaturalist, and if we would see a downward trends of their species based on observations in the community. So my question was, what trends of growth or loss have been shown in the Anaxyrus californicus as shown by iNaturalist? And I just personally thought coming into this that because there are decreasing numbers as the years go by, you would also see decreasing observations within the community. All right, so my methods, I use their expert tool, and I didn't want to use too much information because it wasn't really necessary. In most, I just needed to know where this was found, when this was found, and how many observations were found in what year. Although you can find way more information than this if you choose to use this tool. So I counted the number of sightings per year to know how many sightings were occurring as the years went by. I calculated the increase and decrease throughout the years. And I then calculated the percentage to gather how much of a change there was between the years. So here I have a bar graph to better show, I guess, it's kind of like a better visualization of how you can see what change is going on. And I will get the actual results. So from 2004 to 2005, there's a 50% decrease in the amount of observations. 2006, there was a 100% increase in the amount of sightings. From 2006 to 2007, there was a 50% decrease once again. 2007 to 2008, there was no change. 2008 to 2009, there was, once again, no change. There was no observations between these years. And from 2010 to 2011, there was no change in the amount of sightings. And then from 2011 to 2012, we see a very large increase It was a 33% increase in the amount of sightings. 12 to 13, 70% decrease in the amount of sightings. 2013 to 2014, there was once again an increase. This was a 70% increase, 67%, I'm sorry. From 2015 to 2016, there was a 160% increase in the amount of sightings. 2015 to 2016, there was a 160% increase. 2016, 2017, there was a 160% increase. And then from 2016 to 2017, there was a 400% increase in the number of sightings. So this was a huge increase. 2007 to 2018, there was a 62% decrease, a 2018, 2019 to 20% increase, and 2020, 2019 to 2020, there was once again a 220% increase. So altogether, from the first year being 2014 and the last year that I decided to include, to 2020, there was altogether a 2,700% increase in the number of sightings, 2014 being the earliest year, and 2020 being at the time that I collected the data, the latest full year where there were observations. So this goes against what I was expecting. There was a massive increase in the number of sightings, regardless of the actual population increase or decrease in these species of toad. And actually throughout the years, we see that it's actually pretty random whether there's an increase or a decrease, but altogether, I guess you could say that there's a consistent increase in their numbers. And I thought this would be another great way to visualize this. Most of the space is being taken up by later years with larger numbers. The number of sightings being here 56, 48, 15. And as we go earlier in time, there is way less observations being like 1, 2, or 3. And whenever it's 2012 and above, we kind of see larger numbers actually coming up. So conclusions and discussions. Did I find an answer to my question? Yes, and there was an overall trend of more sightings every year, or more sightings from beginning to end of full years. And it was my hypothesis supported? No, it was not. And my explanation, sightings are never going to be an accurate reflection of species numbers. Also, the fact that iNaturalist and other, I guess, science initiatives like this aren't super well known within the community, so people might not be compelled to share information like this. So public awareness is low, and not many will report sightings, so accuracy of actual sightings and actual organisms being found isn't going to be super accurate. And I think that this would be a great thing to try in the future, or in some point in time where a site like iNaturalist is very popular, or many people are encouraged to use it by, for whatever reason, there's many actually being involved in the community and deciding to report sightings or going out to look for these organisms. We can get more accurate numbers, and we can actually try this again and see if we can see, I guess, if there's any correlation between sightings and numbers. So here are my resources for my research. Thank you.
Thank you very much, Emily. Perhaps you can clap or use the clapping symbol. Thank you very much. We have a few minutes for questions. Who would like to ask the first question? You can use the chat function. You can also raise your hand or just, okay, Lynn has a question. Do you want to ask it yourself, or should I read it out loud? Okay. Any ideas why sightings were so low in 2018?
In 2018, I honestly don't know. If I'm being honest, I'm not sure, but I imagine it would be maybe maybe areas where this organism is commonly found had some sort of maybe temperature or weather conditions that might not allow for people to want to go in and explore areas where they're likely to come across one of these toads, something like that.
Okay, who has the next question in the audience? If there's none, then I will ask a question. Do you have any information about the numbers of toads in these areas? Because sighting, I mean, you could see one and the same toad multiple times, right? So it's not, the data just lists the amounts of sightings, not the amounts of different toads seen, right? They don't control for that.
Control for
how, like whether it's the same animal that is seen.
Like
so it could be that in 2017, like that one toad has been seen by 20 different people, and so there are 20 sightings for one and the same toad. Is that a possibility?
I imagine it could be, but it's not something that really is brought up into question by the website. But I mean, maybe you could go into and see when, like specifically these were cited, and see if there is like specific area or specific location or within that location that one was found. And maybe, I guess you would find that the same toad maybe was found, but since images are uploaded, maybe that can help with seeing if that's a different toad, or, you know, checking the specific location and time and by whom, and that could help you, but that is a possibility. Yeah.
But there's no separate count of the toads. It's just the sightings that they record in the data.
I guess like you have information like who found it, when it was found, country, state, city, image, all these things. So that's as much as, and all the other information, which I personally just didn't check out here, but yeah, it's not like, I guess you mark the toad so you know this isn't a toad you've found before.
Yeah, thank you very much. That's very helpful.
Thank you.
Which other questions are there? Does anyone else have a question? Perhaps Bay or Elizabeth, one of the other presenters, do you have a question for Emily? Okay, if not, then I think we'll thank you again for your great presentation. Thank you very much.
Thank you for everything.
All right, and now can you please stop sharing your screen, and then we will hand it over to Bay, who's going to be the next presenter. Okay, thank you very much. So our next speaker is Beret. Did I pronounce this correctly? Beret? I think you're on mute. Are you on mute?
Yes,
okay.
You pronounced correctly.
Okay, great. Beret is a student at UNCG, a senior, and he's also a full honor student. And his major is biology with a minor in chemistry. And today, Bay is going to talk about effects of potassium perchlorate on developing Meducar primordial germ cells.
Okay. So hi, my name is Barrett, and I'm a student in the laboratory, and I'll be presenting my project, the effect of potassium perchlorate on development with Meducar primordial germ cell. So the perchlorate ions are also genetic chloride compound often used by the military as an oxidizer in rocket propellant and by the artillery, and by industry in numerous other applications such as firework, and for vehicle matches, fertilizer, chlorine products to tobacco, and rocket fuel. It is also a known endocrine-derived chemical and completely inhibits the uptake of iodine into a thyroid tissue in a concentration-dependent manner, and effectively reduces the production of thyroid hormone, lead to hypothyroidism. And perchlorate also is highly water soluble, kinetically inert, and has low absorption tendency, making it a persistent and mobile aquatic contaminant. It has been reported by EPA extract that detection of perchlorate found in a public water system range between 0 microgram per liter and 420 microgram per liter, but the effect of reproductive cells are less understood. Wait, hold on. Okay, an increased number of recently published studies suggest that exposure to perchlorate may cause hematotism, increased angioid level loss testing, reduced sponsor set, increased spermatogenesis, and decreased oocyte duration. However, the effect of perchlorate on primordial germ cell is not clearly understood. So the objective of this project was to better understand the bioaccumulation, taxodynamic, and morphological change caused by perchlorate exposure to epidemiology primordial germ cell and health effect by utilizing Meducar fish as an animal model. We first exposed Meducar embryo to various concentration of potassium perchlorate, 10, 100,000, 10,000, 20,000 microgram per liter perchlorate for 50 days and observed any morphological change on the final day. And we also did co-exposure with vitamin C to see the effect of supplementary have on Meducar fish with perchlorate exposure. And gene expression will observe using RT-qPCR and next generation sequencing was analyzed with, And if figure 2 here show the abnormal change in primordial germ cell migration from primordial perchlorate exposure. Wow. This is a significant increase when compared the 1,000 microgram per liter to the control, with p-value less than 0.05 as shown in figure 3. And that's the abstract right there. One abstract equal to p-value less than 0.005. And we look at the germ cell nuclear antigen gene expression, which protect germ cell from oxidative stress or free radical as PGC undergoes spermogenesis. The expression of GCNA increases significantly in a dose-dependent manner, and we compare the 1,000 to 0 of control. P-value was less than 0.001, which make it very significantly statistic. And this indicates that perchlorate induced toxicity to germ cell. Okay, we also did RNA sequencing and found that the transcriptome activity to represent a non-monotonic graph, as shown right here, U-shape, showing that the 10 microgram per liter and has similar profile as the highest concentrate 1,000 microgram per liter, and overlapping of G between these two show an upregulation as indicated by the red color of my heat map. And we also found area that confirmed that perchlorate does reduce thyroid hormone, as showing a canonical pathway right here. And these are the thyroid hormone gene that were upregulating, that will regulate or deregulating. And we look at the highest concentration, 10,000 microgram per liter and 20,000 microgram per liter. This completely causes death to the embryo, but with co-exposure to vitamin C, mitigate the effect of perchlorate exposure. While vitamin C was able to decrease death, it also increased the depletion of pigmentation of the embryo. And we found significant increase between the 20 microgram per liter and with co-exposure vitamin C when compared to the control. And we also found I don't know why this is blurry, but we also found that the thoroughly signaling pathway to increase in the primordial germ cell. This pathway also protect germ cell from oxidative stress or free radical, maintaining genomic stability and fertility. I have more data than this, but this is the only data that I could fit into this poster. So in conclusion, perchlorate inhibit PGC migration to a genetic ridge and transcriptome analyzing show non-monotonic dose response of perchlorate exposure, and IP analyzing that is our biological software that we use to analyze our pathway, show that the thyroid hormone system is disrupted by PGC due to perchlorate exposure, as shown by the decrease in thyroid hormone. And this study also provide insight into perchlorate effect of future generation, as PGC or primordial germ cell exposure effect have been found to intergenerationally and transgenerationally transmit to offspring across generation. Thank you.
Thank you very much. screen. Oh, it'd be helpful to have still your screen, even if you.
Oh, sorry.
Just for the questions for the Q&A. All right. Who wants to ask the first question about this great presentation? You can use the chat. You can speak up or raise your hand if you like. If not, I will ask the first question. Can you say a little bit more about why you chose this particular kind of fish for your study? Was there some, perhaps I missed it at the beginning of your talk, but can you please say a little bit more about why you chose the Meducar?
Yes, so we chose Meducar embryo as a subject model or animal model because the implantation stage was similar to human embryo stages. So where exposure to chemical, this will provide insight to how it will affect the human embryo as well as the primordial germ cell.
Okay, that's a great reason. And it was a little bit difficult to read the captions of your figures with this presentation. So it would be perhaps helpful to zoom a little bit more. On the right-hand side, the A, B, and C figures, you didn't talk much about those three figures. So what are they representing?
So these are the genes.
Oh, no, on the right-hand side, the A, B, C,
this.
No, on the right hand. Sorry,
right side.
Sorry. For me, it's the right. I'm sorry. I guess for you should be also the right.
Yeah, no, my bad. It was my fault. Yeah. So these are the genes that were regulated by the thyroid hormone.
Mm-hmm.
So right here, the blue, my legend right here, like predicted inhibition of thyroid hormone, means that this is decreased the thyroid hormone, and this can lead to a decreased measurement of this gene right here, as indicated by my legend.
Oh, I see. Yeah, that's helpful. And the differences in ABC, can you please say a little bit more about the differences between these three different?
These three, A, B, C, and D. A was the 10 microgram per liter,
Mm-hmm.
and B was the 100 microgram per liter, and C was the 1,000 microgram per liter.
Oh, I see. Okay.
Yeah. So as you can see, when you compare A and C, you can see more gene be regulating, showing a similar profile with the lowest concentration, the highest concentration. But with the medium concentration, you see low gene expression, low G found to be regulating.
Mm-hmm. Okay, who wants to ask the next question in the audience? Perhaps one of the other presenters, would you like to ask a question about this presentation? We still have plenty of time for talking about this talk. Still have 5 more minutes. Chris, you want to ask a question?
Yeah, thank you for Bae. That was a fantastic presentation, and it's actually really helpful to zoom in because I can see some of the details that I couldn't see before. So it's really nice. It's also I'm impressed by the depth of analysis here and the specificity. I will also say, as having a non-scientific background, I was struggling to understand a lot of what you were the details you were saying. I wonder if there's a way that maybe you can put what is the impact of this research for someone like me who doesn't have a scientific background. How might I understand the impact of the research in a common way? Okay,
so with soldier, join the military, they're not, they don't know that they're supposed to have concentration of chemical, like the artillery that they use it, the gun, the rocket that's being used by the military, the soldier, they don't know that, chemical is found in those, material. So we found that perchlorate is a highly concentrated chemical found in the military, the material, the gun, those kind of stuff. So we want to see how this chemical which is found in the environment and is being man-made by such as firework, airbag knife for vehicle matches, this kind of stuff, and be released to the atmosphere and environment. And we want to see if how this chemical perchlorate have effect on the soldier and people around exposed to perchlorate. And so we want like specifically look at the primordial germ cell or the germ cell that begin to develop to sperm, because sperm is like can be generally inherited to offspring. So if anything like happen to the sperm or germ cell, the offspring will also have the same effect as the parent as well, like the epigenetic methylation, like epigenetic pattern. So we did a next generation sequencing or library preparation, RNA sequencing and gene expression to look at what gene in the primordial germ cell that was affected. And we did numerous like gene, but I was only able to present gCNA because it was like the main gene that protected germ cell. It imitate the genomic stability and fertility for the adult to able to fertility or produce offspring. And if perchlorate like causes a decrease in gCNA, this will mean that the person or the male will not able to like produce sperm like fertility, what's called sterile sperm or sperm that able to like fertilize. And then we also found that the primordial germ cell, the germ cell, didn't migrate to the area or the gonad where it's supposed to develop into sperm. It was like the primordial germ cell was stuck in the abdominal right there, like the embryo. So it didn't have like, most of them or majority that did like migrated to the gonad to develop to the sperm. That causes a problem to fertility because you need sperm or primordial germ cell in the gonad area to produce, fertility or offspring. And then when we did the RNA sequencing or next generation sequencing, that will provide insight into how it will affect the offspring or next generation. We found various gene, and then we found that the 10 microgram per liter, the lowest concentration, and the 1,000 microgram per liter, the highest concentration, has similar profile. Because usually highest concentration like causes like an increase or decrease significantly. But in this case, because perchlorate is an endocrine disruptive chemical, that means that it's very unpredicted to the human body. So we showed that the 10 microgram per liter or the lowest concentration perchlorate causes the same effect as the highest concentration perchlorate. So that created like a significantly problem to human health, because usually we think that the lowest concentration of chemical doesn't have any effect, but it does because our body get tricked our cell get tricked that because it's the lowest concentration, it will not do anything. So that's how it able to bypass like all the regulatory part of our cell, and they're able to suppress G or express G significantly. And then perchlorate, where I encounter article, perchlorate is known to decrease the thyroid hormone, for various articles related to perchlorate. So we want to see if we want to confirm if perchlorate do cause decrease the thyroid hormone, and we do found evidence and confirm that perchlorate does decrease thyroid hormone, as shown in our IPA analyzing for our RNA sequencing. The RNA sequencing showed the gene expression or transcriptome activity or gene level when compared to control when you're screening. And then we also did co-exposure with vitamin C to see if the nutritional supplementary that we take every day could help our health. So we use vitamin C, 1 milligram of vitamin C, co-exposure with perchlorate. And we found that it significantly, or not significantly, but it highly reduced the mortality rate caused by perchlorate. But at the same time, we also found that vitamin C causes an increase in depletion to the pigmentation of embryo. Not only this shows that vitamin C is helpful, but at the same time, it also like causes side effect. And from our RNA sequencing data analysis, we found that this pathway was altered. So the significant of the main pathway that was related to germ cell was the certain synergy pathway right here. So this pathway, or the gene that is involved with this pathway, is also protected germ cell to the gonadal area to develop into sperm. And then all of these other pathways relate to endocrine disruptive chemicals that have effect on human health, or the effect of endocrine disruptive chemical. So in conclusion, this study provides insight into how it will affect the person at the moment and the future generation from their germ cell. So this study was very important, and it has been submitted to a professional journal. Yeah.
That's great. Congratulations.
Thank you, Bay.
Thank you for this many helpful insights into your research. Thank you very much. Unfortunately, the time is over now for your presentation, but thank you again, and good luck with the journal submission.
Thanks.
We're now going to turn to Elizabeth's talk. Those, do you want to try sharing your screen with us? All right, so Elizabeth will going to be presenting the last paper for today. She's a junior at East Carolina University and has a double major in biology and Hispanic studies on the pre-med track. She's also an honor student at the East Carolina University, and she has already quite a bit of research experience under her belt. She conducted research at the Duke Clinical Research Institute, and she's also working for the East Carolina Diabetes and Obesity Institute. So that's pretty impressive for a junior. So I look very much forward to your talk, which she's co-presenting or presenting for her team. Other members of her team, Valerie Jurgis, Vincent Kindulio, Emma Kwambia, and Jenny Yan. How I pronounce those names correctly. If not, please correct me.
Yeah, thank you. That's a good intro.
Okay, so you're welcome to start now.
Thanks. And thank you for the introduction. My name is Elizabeth Chan. My teammates unfortunately had schedule conflicts. I'll be presenting our research. We conducted our research through the Duke Summer Training and Academic Research Program at the Duke Clinical Research Institute this past summer. And we worked with our mentor, Dr. Ryan Kilpatrick, who is a NICU fellow at Duke. And today I'll be presenting our research on urinary tract infection epidemiology in U.S. neonate intensive care units. I'll be defining urinary tract infections, our neonate population, and risk factors. I'll also discuss our methodology, our findings, and reflection of the study. And first, I'll give a background information on urinary tract infections, neonates, and risk factors. So UTIs are the second most common infection amongst children. They're defined as a bacteria or yeast-caused infection in the urinary system. And the UTI symptoms include fever, irritability, and vomiting, and the effects of UTIs are more severe in infants as their immune systems are not yet fully developed, especially in the case of infants who were born premature. There are a few ways to diagnose UTIs, but the most common is through a urine culture, which is a lab test that checks for bacteria, yeast, or other microorganisms in the urine. And for neonatal infants, the positive result is a culture growing at least 50,000 colony-forming units per milliliter from a single organism. There are two ways to collect sterile urine to test, and the first is a suprapubic tap, a procedure involving an insertion of a small needle directly into the bladder to collect urine. And then the second is a urethral catheterization, which is a procedure that uses a catheter, which is passed into the bladder to allow urine to drain. For the purpose of our research, neonate or infant is defined as a patient who admitted to one of the 367 NICUs reporting data to the pediatrics database who were born at less than or at 32 weeks gestational age, as well as that they were in the hospital past postnatal day 7. In addition to this, our study focused on very low birth weight infants who weighed less than 1,500 grams at birth, and this category also includes extremely low birth weight infants who weighed less than 1,000 grams at birth. And for your reference, 1,500 grams is a little bit less than 3.5 pounds, so very small babies. When looking at risk factors for UTIs, we defined risk factor as any variable that increases or decreases the chance of developing a disease, in our case, a UTI. The risk factors we observed were sex, birth weight, gestational age, prenatal steroids, and lastly, race and ethnicity. And for our methodology, we identified all infants admitted to 367 NICUs in more than 30 states and Puerto Rico that are managed by the Pediatrics Medical Group. For this study, all infants within the Pediatrics Medical Group from January 2008 through December 2018 were considered eligible. And then we refined our study population with only very low and extremely low birth weight infants born at less than or equal to 32 weeks gestational age and hospitalized past postnatal day 7 were selected. Infants born at a non-pediatrics facility and then transferred to one were excluded, and those born with extreme abnormalities were also excluded. Stata was used for the statistical analysis, and p-values were also calculated for unadjusted comparison of the two groups using either the Fisher-Exact test or a chi-square test of association for categorical and t-test or Wilcoxon rank sum test for continuous variables. Multivariable logistics and Poisson regressions were used to evaluate the unadjusted odds of UTIs, normalizing the infant's weights to the typical weight for gestational age. And from the final model, odds ratios of 95% confidence intervals were reported as well as predicted probabilities. Our findings show several factors that impact UTI risk in neonates, including sex and circumcision, birth weight and gestational age, race and ethnicity, and lastly, prenatal steroid exposure. Regarding the sex of an infant, as seen on the table, females from our study population were assigned an odds ratio of 1 in order to compare the male risk of contracting UTIs to the females. And then the data shows that the males had a significant odds ratio of 2.05, which means they're more than twice as likely to contract UTIs. In addition, males accounted for 68% of UTI diagnoses, despite only being 50% of our study population. Not being circumcised is associated with an increased risk for UTIs, as the presence of foreskin correlates to a higher amount of periurethral bacteria. And circumcision has been linked to a decreased risk of UTIs in male neonates. However, most premature infants are too small and too young to be circumcised, and this explains why the males in our study were twice as likely to be diagnosed with a UTI compared to females. Since male neonates are too young to be circumcised, we recommend monitoring males closely and collecting urine cultures immediately after any symptoms are noticed in order to prevent damage that a UTI might cause. If the urine cultures provide a positive diagnosis, then swift treatment should be started. For birth weight, infants weighing 500 to 999 grams represented 61% of the UTIs in our study and had the highest unadjusted odds ratio of 1.14, which meant they were 14% more likely to contract a UTI. And this population might be small enough to have a weakened immune system, but big enough to survive to the point of infection. And thus, there may have been an increased chance of contracting a UTI. Alternatively, the elevated mortality rates for the smallest and youngest infants may translate to an underdiagnosis of UTIs in this population. When we looked at gestational age, infants less than 24 weeks old were most at risk of UTIs as compared with those at other age groups. And this might be a result of a less defined immune system of younger neonates. However, after we normalized the infants' weights to their gestational age, infants that were small for their gestational age were less likely to have a UTI diagnosis with odds ratio of 0.78, and this was unexpected because we thought that the smaller the premature infant, the more susceptible they would be to contracting UTIs. So this might be due to SGA infants having weaker immune systems, which leads to a weaker immune reaction and less severe symptoms, which could lead to a decreased likelihood of receiving a urine culture in the first place and being diagnosed. Thus, we recommend that SGA infants be closely observed and considered for empiric antibiotic treatment. Regarding race and ethnicity, infants identified as Hispanic were associated with a disproportionate number of UTI diagnoses with 14% higher incidence than other groups. and in infants identified as other also had higher incidence. However, we don't think this is a result of any racially ascribed differences. It's more likely that this variability in prevalence is due to regional differences in clinical practice or collection methods in places with different demographic makeup. Some hospitals might collect urine culture more often than others, and some have a higher or lower threshold for how soon to test for UTIs. So it's these differences that we suspect contribute to the higher UTI diagnoses in the Hispanic patients observed. When a healthcare provider knows that an infant will be born prematurely, they may prescribe steroids to strengthen the baby's lungs. So in our study, of the infants whose mothers were treated with these steroids, we found an increased prevalence of UTIs. Infants with prenatal steroid exposure had a significant odds ratio of 1.13 compared to infants without prenatal steroid exposure. And this finding is novel, and we believe it should be further explored. However, we suspect that prenatal steroid exposure may vary UTI risk because these infants had a better chance of initial survival due to their stronger lungs, which may give them more time to contract UTI as compared to infants that didn't receive prenatal steroids. So we recommend that infants whose mothers were treated with prenatal steroids be given additional consideration when a UTI is suspected, and if necessary, they should be considered for empiric or preemptive UTI treatment. We also studied which agents cause a positive UTI diagnosis and found that E. coli and Enterococcus were responsible for the biggest portions. Something notable was the presence of Candida as the 5th most common causative agent, and this is significant because Candida is a yeast and the others are bacteria. Due to premature infants' weakened immune system, they oftentimes won't present any UTI symptoms or show very nonspecific ones. So UTIs can be extremely severe in this population, so a physician might want to start an infant on a wide-ranging course of antibiotics, since waiting can have detrimental effects to their health. However, antibiotic resistance is growing fast, especially to common bacteria like E. coli, and common antibiotics are losing their effect. As such, we recommend that healthcare professionals exercise caution when treating UTIs with antibiotics, as antibiotic resistance continues to become more common. So now I present the importance, limitations, and advantages in future research. So through this research, we gained knowledge on the correlation between certain factors and risk of contracting a UTI, and this allowed us to make recommendations to benefit the health and well-being of patients in NICUs. Despite our significant findings, our study also had some limitations since we didn't recover data on the mode of urine collection, and we didn't have the odds ratio for some of the secondary risk factors. However, analyzing data in infants from over 300 NICUs provided an advantage, as most neonatal studies are often single-centered, and this allowed us to mitigate regional bias and increase representation in our study population. Our study also bolsters literature available on UTIs in very low and extremely low birth weight infants, as there was only a minimal amount of study focused on this topic. So further research is necessary to verify our association between these factors, small for gestational age infants at increased UTI risk, prenatal steroid at increased UTI risk, and race and ethnicity and varied UTI risk. There's not much prior data available in the literature corroborating these findings, so we encourage future exploration to test their validity. So thank you everyone for attending this presentation. I also like to give a special thanks to our mentor, Dr. Ryan Kilpatrick, and our principal investigators, Dr. Benjamin, Dr. Danny Benjamin, and Dr. Kenesha Zimmerman. Thanks.
Thank you very much. This was very interesting. Who would like to ask the first question? You can post your question in the chat. You can raise your hand or turn on your video. If not, then I will ask the first question. So you talked about the race impact, and as far as I understood you, you said that you don't think it's a significant difference. Can you say a little bit more about this part of your study, what was the ratio of white and non-white babies in your study, and why do you think that this difference is not significant?
Yeah, so that's a really good question. We were really confused about that at first. So of the positive diagnoses, the majority, we had 37% white, 30% Hispanic, 26% Black, and then 5% other. So we did think this was really strange that they weren't evenly spread across, because there aren't any anatomical differences that would lead to increased prevalence of UTIs. So then from this, we thought that maybe the higher incidence is due to more regional areas in different hospitals, because some hospitals have different protocols for how soon they treat, or how soon they take urine cultures. And then it could have just depended on which hospitals report to the pediatrics database. There could have been in areas where there's a higher Hispanic population as well.
Okay, but you think that the difference is like not significant? It's just a more random outcome of the collected data.
Yeah, I wouldn't say it's more like it. I wouldn't say there's an increased UTI risk depending on what race or ethnicity you are. I would say it's just happened to be that way because of maybe how they report data or the location of the hospitals.
Okay, thanks. That makes sense. So then we have Chris in the comments says, first, very good job on the research and presentation, very interesting. And then he says he has to go. Oh, sorry, he just told me that he has to go. Okay. What other questions are there? Bei, do you want to ask a question? If not, I can ask another question. Bei, can you hear me?
Yes, no question from me. She did really great. Good.
Okay. So when you ran your analysis, were you part of collecting the data, or were you given the data and you did the statistical analysis and the qualitative analysis of the data?
Yeah, so it was through this Duke Star program this past summer. So we worked with statisticians to help go through the statistics. So my teammates and I more so did the qualitative analysis of like why odds ratios were higher or lower.
Okay, yeah, that's like in that program. So you were a bigger team of statisticians and medical experts, and then your team, and you jointly work together on this project. And I figured it's part of is a part of a bigger project on NICU babies and the urine infections, or was your group the only one focusing on this aspect of the NICUs?
My group was the only group focused on it. The way that Duke Star worked was there were 4 groups and they all had very different topics, but ours was on UTIs in NICU patients. But yeah, we had my teammates, the 5 of us who are undergraduate students, our advisor who's a fellow, and then our co-principals who are faculty in the Duke Clinical Research Institute, as well as a statistician.
And the other teams did also research on the same data, but with different topics? Or you had the same data pool, or were there 4 separate data pools and 4 separate projects?
They were completely different data pools.
Okay.
So like some of them did research on like vaccinations during COVID and other.
Oh, I see. Yeah. So that's very helpful. Okay, the time for our session is up now, so I think we have to stop. But this is very interesting, and thank you very much for sharing your research with us. I hope you can continue doing something like that, because it seems like you got a talent for it, and I wish you all the best for your future research. And thank you very much, for sharing your research with us.
Great, thank you.
Okay, the next session I believe starts at 12. If you want to join any of the other sessions, I think the next one is at 12 to 12:50, and then the presentation of the awards is at 4:30. Okay, have a great rest of your day.
Bye.
Bye.